Good morning. Today we will study about Lac Operon. So basically, what is Lac Operon? It's a gene regulatory system present in E. coli, Escherichia coli bacteria. For what purpose? To digest the lactose, mainly breakdown of lactose into glucose and galactose. So, why we need Lac Operon? To save energy. How? The main principle is when the substrate, that is the lactose, is present, operon is on. When the lactose is absent, operon is off. So now we will see what happens in absence of lactose. So we will first draw the gene, the portion of our DNA. Sorry. So now I will describe the structural components of this gene. So this is basically LAC-I, it's a regulatory gene, and this is the promoter for this regulatory gene. So this. Now this is operator. I will describe this function later. Then we have promoter 2, which controls the activity of these three genes, LAC-Z, LAC-Y and LAC-A. So basically what happens, this is a regulatory gene which synthesize repressor protein. So now this repressor protein has two binding site. Okay, two binding site. Suppose I will first talk about this site. This site binds to the operator region. So now what happens? This goes here and binds to the operator region. And when it binds to the operator region, it blocks the binding of RNA polymerase. So the RNA polymerase present here, one second, cannot bind to the promoter region to start the transcription, translation and the enzyme production, which further helps in the lactose breakdown. So now what happens if lactose is present? Now if lactose is present, फिर क्या होता है? Then एक सेकेंड, मैं इसको कॉपी कर लेती हूँ. कॉपी. When lactose is present, this gene synthesize the repressor protein. Okay, in the same way, it has two binding site, but the lactose here converts into allolactose with the help of beta galactosidase enzyme. So now these allolactose attach to the second binding site of the repressor protein, and when allolactose is attached, that is, this become inactive repressor. Okay, so now this cannot bind to the operator site for the RNA polymerase gets open, and the RNA polymerase binds to this promoter region, which further helps in the transcription and translation of the LAC-Z, LAC-Y and LAC-A gene. So through transcription and translation they produce the protein. So basically their enzymes. LAC-Z produce the beta galactosidase enzyme, which further helps in conversion of lactose, that is breakdown of lactose into glucose and galactose. And the LAC-Y gene produce the permease enzyme, which help in entry of lactose into cell. And the LAC-A gene produce the trans-acetylase enzyme, and the function of this enzyme will be studied later in the higher semester. So basically this is the transcription and translation process. Now these are the main funda for this breakdown. We have gone through all these steps. Now a key point, that is, this is called operon. Why? Because the multiple gene LAC-Z, LAC-Y, LAC-A are controlled by a one single promoter. So why it is called operon? First of all, why operon? So the answer is one single promoter controls the multiple, here 3 genes. Okay, now why it is called negative and inducible operon? Negative cause the repressor protein present here gives you the negative effect, because when it binds to this site it blocks the binding of RNA polymerase and further transcription translation process, and when it can't bind it allows the transcription and translation process. So this repressor protein gives the negative effect. And why it's inducible? Because it is induced in the presence of lactose, so it is called inducible operon. So now, okay, my point is, when the lactose is absent, if no enzymes are produced, then how lactose enters the cell with the help of permease? So basically the answer is, when the lactose is absent, all the transcription translation process occurs, but in a very low rate. Transcription and translation occurs at very low rate, and all the enzymes are produced at a very less amount. Okay, so less amount of enzymes produced here. So this is basically the basic Lac Operon. Now we will study about a special case which is called the catabolite repression. So it is basically controlled by glucose. When glucose is low, it produces CMP-CAP complex, and this complex binds to the operator region of the gene, and it reduces the efficiency of binding of repressor protein. Hence, it induces transcription and translational process. And obviously the glucose is formed because the glucose amount is low, so the glucose is formed. But when glucose amount is high, the CMP and CAP complex not formed, so obviously can't bind to operator region, so repressor protein can bind effectively to operator region, and the transcription and translation process slows down and the glucose not formed. Glucose not formed, and here the glucose formed. So basically why it is called repression? Because when it's low, the transcription translational rate are high, but when the glucose is high, the transcription translation rate is low. So in the presence of the glucose, okay, that is product, the whole process is repressed, so it is called basically the repression. So here it is all about the Lac Operon and its special case.